Hello everyone, welcome to ITLS Academy. In this session, we will be starting with the topic of medical biotechnology. So, this is a module of some of the most prominent topics of medical biotechnology. And starting with the first session, the first topic that we will cover is fundamental techniques in medical biotechnology. So, part one, because one session will not be sufficient, so we will be continuing in the next session. So, before going into the details of fundamental techniques, I just want to give a very brief and concise introduction about what is medical biotechnology. So, medical science, we all know, we all have a rough idea about what is medical science. So, any science which is dealing with the human body, which is dealing with the medical perspectives of the human body, is what is medical science. And what is biotechnology? Biotechnology is technology dealing with biological sciences. So basically, when we are clubbing the two words medical and biotechnology, the the brief idea or the fundamental concept of medical biotechnology is it involves the different applications of technology uh, related to biological science and medical science. Okay, and mainly focusing on medical science, hence medical biotechnology. Okay, now, starting with the main topic of this session. So, as you can see over here, I will just highlight detecting and diagnosis human diseases condition. Now, why is it important? Because whenever we are talking about medical biotechnology, we are dealing with the human body, and human body means human diseases, the different diseases that can happen in the, inside the human body. Okay, now particularly again when we are focusing on the medical biotechnology aspect, we will be mainly focusing on the different genetic diseases that can occur inside the human body. Okay, and this table, as you can see, the table that I have given over here, this mainly focuses on the various genetic disease conditions that can happen in the human body or which are usually reported to occur in the humans. Okay, so among them, one of the major or most common genetic disease that can happen is cancer, as we all know. Now, talking about cancer, cancer is genetic, but also there are some viral cancers also. Okay, but it is thought about, or it is it is believed, there's a hypothesis that each and every type of cancer, whether it's viral or genetic, has a genetic predisposition. Means even if it's a genetic cancer, then the obviously genes are the reason, genes are the culprit. But also if it's a viral cancer, in that case also there can be a genetic predisposition, or there has to be a genetic predisposition. That is the concept, okay, given by many researchers. Second important genetic disease, as you can see over here, is cystic fibrosis. Okay, now this is again a genetic condition which is which mainly leads to a respiratory distress syndrome. Okay, so it basically goes and affects the lungs. Then we have something called muscular dystrophy, where it will lead to various muscle paralysis conditions. Okay, then we have hemophilia, which is a blood disorder, genetic blood disorder. We have sickle cell anemia, which is again a genetic blood disorder. Then there are some diseases like phenylketonuria, Tay-Sachs disease. So these are basically metabolic defect of enzymes. Okay, so we call it inborn errors of metabolism, because it happens due to the genetic defects in the genes which are supposed to play an important role in the genetic defects in the enzymes which are supposed to play a very important role in the metabolism. Okay, we all know that enzymes are a key factor as far as metabolism is concerned. Okay, so moving forward, detection of genetic diseases is also a very important fundamental aspect of medical biotechnology. Okay, so now whenever, since we are focusing on genetic abnormalities mainly because we are talking about medical biotechnology, so there has to be some kind of technology, isn't it? So the screening of the genetic disorders is something very important. And talking about the screening or the detection part of genetic diseases, it's very helpful, or it's very beneficial if we can detect the genetic disease before the baby is born. So that is what is called prenatal screening of genetic disorders, which is done by various methods. And over here I have mentioned two most prominent methods, as we can see in the diagram also. You can I will be explaining. So one is chorionic villus sampling and the other one is amniocentesis. Okay, and both these two types of testing of genetic disease will lead to the karyotype. Okay, now what is karyotype? Karyotype is basically where we can get a picture of the different genes as distributed in the inside the body of that particular patient or that particular person who is suffering from that disease. Okay, so chorionic villi, what is chorionic villi? It is basically the covering, as you can see over here. It is basically the covering of the, it's a part of the placenta, which is very much important for any kind of nutritious exchange or gaseous exchange between the fetus and the mother. So if we can just collect a small amount of sample from that chorionic villi, then we can go for testing of the genes. Okay, so that is one method which is usually carried out at around 8 to 10 weeks of pregnancy. And another technique that we can follow is amniocentesis, where we are actually collecting the amniotic fluid. What is amniotic fluid? It is again a fluid which is accumulated inside the amniotic cavity of the mother of the womb. Okay, cavity formed by the the placenta inside the womb of the mother. So if we can directly take out a sample of that amniotic fluid, which is known as amniocentesis. Okay, so special type of needle is actually used. Okay, it's a ultrasound guided procedure, USG guided procedure. So that can also be done, and then we can uh, get a nice picture of the karyotype of the baby, and from there we can study if any kind of genetic disease is there inside the uh, baby. Okay, now moving forward, there are other techniques also, like again one of the most prominent techniques will be FISH, that is fluorescent in situ hybridization. Now what is in situ? The word in situ means where we are mimicking the exact conditions, actually not mimicking, where we are performing the procedure or performing the experiment in the natural conditions. Okay, so it's not that we are performing it in inside the lab, or we are mimicking a condition exactly like the body and then performing it somewhere else. No, it's not like that. So in situ means in natural conditions, in original conditions. Okay, and hybridization means where we are hybridizing something with some other compound. That is hybridization, as we all know. And what is fluorescence? Fluorescence, we all know what is the phenomena of fluorescence. So we are basically using the fluorescent probes, and they are, probes means what? Probes means which are specific to some sequence or specific to some chromosome or gene. So say for example, if I want to detect a particular type of genetic disorder, and I know that this genetic disorder occurs due to defect in this particular gene, so I will develop a probe against that particular gene, and that probe will be fluorescent. Why fluorescent? Because in that case it will be able to give us the signal by the color change, exactly what it is shown over here, as you can see in the diagram. Okay, so we have the rRNA, and that is, say for example, our target gene, and then we have the fluorescent dye which is being loaded with the probe. So accordingly there will be a color change, where when we will be observing it under the fluorescence microscope, it will produce a specific type of color. And if that color is produced, that means it is positive, that particular gene is present in the patient. And if the color is not produced, if there is no fluorescence, then we will conclude that the result is negative, means the patient is not suffering from that particular genetic disorder. Okay, uh, another important technique will be SNPs. Now what are SNPs? The full form is single nucleotide polymorphism. Okay, very important. Now, what is single nucleotide polymorphism? It is one of the most common forms of genetic variation, and it is estimated that one SNP occurs approximately every 1000 to 3000 BP in the human genome, which is again a very, fascinating characteristic of SNP. Okay, and also 99% of the DNA sequence that we have will be exactly the same. Means 80% of 0.1% variation will be SNPs. मीन्स जो 0.1% वेरिएशन रहेगा उसमें 80% will be SNPs, means single nucleotide polymorphism. Okay, so again over here you can see a diagram. So this is person 1 is having a normal protein. Okay, so some DNA variations have no negative effects on protein structure and function. Means normal protein means key, even if there is DNA variation, it has not mattered to that person. Okay, but in case of person 2, we can see there is a SNP. there is a single nucleotide polymorphism, as a result of which the protein that is formed, because we all know the the main concept or the basic concept of genes must be very clear, and that is the central dogma. What is it? From DNA to DNA, that is replication. Then from DNA we will have RNA, that is transcription. And finally from RNA we will have protein, that is translation. So that is the central dogma, and this concept is very important. So whatever gene we are talking about, no matter what type of genetic disease it is, whatever gene we are talking about, it's very important to have the clear concept that a gene is responsible to give rise to a protein. Okay, so if a protein is not functionally active, if a protein is not biologically active, that means the gene that was responsible to code that protein is not right. Something is wrong with that gene. Any kind of mutation can be there. Maybe the gene is not there only, or maybe the gene is overexpressed or very much underexpressed. Okay, any type of condition can occur. There are various types of genetic mutations. I will be talking about them in the upcoming sessions, no need to worry. So that is the main concept. Okay, so if there is a type of SNP, then the protein that is formed will not be functional. Okay, so either it will be malfunctioning or non-functioning, or the functionality will be at a low range, low level. Okay, moving forward. So another technique that we can go for to diagnose or detect genetic disease will be microarray analysis. Okay, so microarray needs to be created containing some disease genes or SNPs, and then DNA from the patient is stacked with the fluorescent dyes and it is hybridized to the chip. So basically it's the same concept like fish, because here also we are using fluorescent probes, just as we were using in case of fish. Okay, and then binding of a patient's DNA to a gene sequence on the chip will indicate that the person's DNA has a particular mutation or SNP. So basically it's the same thing, that if there is proper binding with the fluorescent probe, and if there is a color change, there is a signal of color change, that means that particular gene is present. Okay, so over here also I have given a diagram, so it's kind of the same thing. So first we need to know the the family history, the entire details of the the genetic abnormalities, or if any kind of genetic disease was present in the parents. So all those medical history basically that we need to take. Then we need to have the cell sample, because from there we need to collect the DNA. So after collecting or isolating the DNA from the sample, we have to prepare the microarray using the disease gene or the SNPs, and then we need to summarize or take the genetic profile of the patient. Okay. Now coming to something called protein arrays. Now what are protein arrays? Same thing, but here instead of using the DNA or the genes, we are using the protein. So protein means basically antigen. Antigen, antibody, both of them are protein in nature, we know, and they are antagonistic to each other. Means the antigen will go and react with the antibody depending on specificity. Obviously, there needs to be a particular antigen binding region, a particular antibody binding capacity. Okay, I will be talking about again in details about antigen antibody in the upcoming sessions. So here basically we need to collect blood from the patient, because in the blood only, in the serum only, we will get the antigens or the antibodies. Then we need to design a particular antigen or antibody, vice versa, and we need to load or what to say, we need to fix that antigen or antibody with the antibody, precisely with the particular probe. And in this case, as you can see, the probe is a color producing enzyme. Okay, so either it's a fluorescent probe or it's a color producing enzyme, because when we are visualizing, when we are trying to interpret the result, it's very important that there is a kind of change which we can detect visually, which we can detect qualitatively. Okay, and what better than seeing any kind of color change, isn't it? So that's why we always prefer fluorescent probe or a color producing enzyme. Okay, again moving forward. Now coming to a very important concept, that is pharmacogenomics. Okay, now what is pharmacogenomics? The word pharmaco deals with drugs, as we all know, and the word genomics deals with genes, as we all know. Okay, So pharmacology is a branch of study in case of medical biotechnology where we are dealing with various types of drugs, particularly the newly evolving or the emerging drugs. Okay, so they can be cancer drugs, they can be drugs against infectious diseases like COVID, SARS. Okay, so newly upcoming emerging drugs. And in that case, it's very important that we also have a fair amount of idea about the genetic makeup of the patient. Okay, because no matter what kind of drugs, or no matter what kind of condition, no matter what kind of extent of the disease a particular person is facing, even if we take, say for example, we have a sample size of 10 patients, and we are administering the same drug to all those 10 patients, because obviously, say for example, all of them are suffering from pneumonia. Okay, but if we observe precisely, if we examine each of those 10 patients very precisely, we will see that each of them are responding to the same medicine, to the same drug, which is obviously in this case an antibiotic, since we are talking about pneumonia, it's a bacterial disease. So each of those 10 patients will be responding to that same drug, to that same antibiotic in different ways. And also they will be having different side effects. Why? Because of the genetic makeup. Okay, although we are like almost 99.99% genetically identical to each other, because we are the same species obviously, but still that 0.1%, 0.01% difference makes it, makes the difference. Okay, so it is very important that we study the genetic makeup of the person before prescribing a drug, and that is what is pharmacogenomics. So pharmacogenomics basically deals with the concept of personalized medication. Okay, so that exactly what I am showing you in the next slide, as you can see. So see, personalized medicine. So what they are showing over here, see, this is a group of cancer patients and we are giving a medicine, and we are seeing that in this group all of them are suffering from the same cancer, same type of cancer. Say for example, breast cancer example we have taken here, and the main gene which is responsible for like the main mutation of the gene that is responsible for causing breast cancer is BRCA1 or BRCA2 gene. Many other genes are also there, but the most prominent is this one. Okay, so we are seeing that if we carry out the same chemotherapy, chemotherapy means cancer therapy, a particular type of cancer therapy. So some of the cancers, they need oral chemotherapy, some need IV chemotherapy, depending on the dosage of the drugs, depending on which type of drugs we are using, and also depending on the condition of the patient, how much the cancer has spread inside the body of the patient. So these are several factors that we need to think about before deciding about whether to go with oral chemotherapy or to go with IV chemotherapy. Also there can be radiation therapy, and there can be surgical options also for cancer treatment. Okay, so here we are mainly focusing on chemotherapy, as we are talking about personalized medication. So some have showed very good results, some have shown no effect, means they have shown no response to the treatment, neither good nor bad, while only one person, as we can see over here, has shown a very adverse effect, means instead of recovering, instead of becoming better, that person's condition has worsened. But see, very careful, we are using the same medication, we are using the same therapy for all the patients, and all the patients are suffering from the same type of cancer, that is breast cancer. Okay, but in this case, just come to the right hand side of the diagram. In this case, what we are doing, we are isolating the DNA, we are isolating the gene, and then we are studying the gene, and we are seeing that the, if we are like on the basis of the study of the gene study, if we give different drugs. Okay, so to this three people who have shown a good response, we are giving the same drug. But these bunch of people who have shown no effect, neither good nor bad, we have come up with a different type of drug. So see, now they are having a good effect. And on this patient who have had an adverse effect, we are again modifying the drug and giving something else. So that is precisely the concept of personalized medication, where we are depending on the genetic makeup, depending on the genetic like uh, institution of the patient, we are modifying our drug. Okay, next important topic will be how to come up with personalized medication, because we need to modify the same drug, isn't it? It's not that we are coming up with a different type of drug, we are just doing some minor structural modifications of the drug. Okay, so that can be done by drug solubility, drug breakdown, or drug elimination. So these are the three types of techniques that we can go for if you want to influence, if you want to better, or if you want to do some kind of modifications or exploitation of the drug's capability or effectiveness. Okay, and also we can come up with something called microspheres. What are microspheres? These are basically tiny particles which can be filled with drugs. And why they are important? Because they can be very much effective in delivering the drug to the precise location. Okay, because whenever, so this is a nice diagram which is basically explaining. So we can just take a spray bottle which will be containing those microspheres, and inside those microspheres we will have the drug, the chemotherapeutic drug. So basically these microspheres will be acting as the carrier molecule. Okay, also in this respect, when we are talking about carrier molecules, nanoparticles are also very important. Okay, what are nanoparticles? Nano means very tiny or very small. Okay, uh, obviously they cannot be seen with our naked eyes. Okay, so nanoparticles are precisely or mainly they are made up of metals. Okay, we have zinc nanoparticles, we have lead nanoparticles, we have mercury nanoparticles. Okay, so basically they are like constituted of metals. So they can be used as effective carriers when we are talking about drug delivery, because in case of cancer it's very important that whatever drug we are giving, whether it's oral or IV or whatever chemotherapeutic, whatever anti-cancer agent it is very important that its delivery inside the body is very precise. Means what? Means it's very important that the drug will be able to go and attack or exert its function on the specific cancer cell only. Because otherwise, in most of the cases, what happens in most of the cancers, why the recovery rate or why the cure chances are not that much high? Because the cancer drugs, they don't know where to go, they don't know which cell to target. They target all the cells of the body, and that is one of the major reasons behind the side effect of chemotherapy, that is hair loss and also nails are also gone. Why? Because this hair and nails, they're very actively or highly dividing cells, fast dividing cells. And the main property or the main feature of these anti-cancer drugs is that they attack the fast growing cells. They only know that we need to go inside the body and attack the fast growing cells, because that is the main feature of cancer, any type of cancer. What is cancer? Uncontrolled proliferation of cells, uncontrolled proliferation or uncontrolled multiplication of cells. Okay, so in order to stop that, we are giving the chemotherapeutic drug. So the drug knows only this much, that inside the body, as soon as I go inside the body, I will start attacking the all the first dividing cells of the body. So that includes our hair, our nails also, but they have done no harm. So for example, if a person is suffering from breast cancer, her hair or her nails, they do not need to die, isn't it? They do not need to be killed, but the breast cancer cells need to be killed. So in most of the cases, what happens, the drug doesn't know which cell to particularly attack. It doesn't know how to recognize its target cell. So it just rampantly starts to kill all the fast dividing cells of the body, which can be very harmful. And as a result of that, we are having the different adverse side effects of cancer therapy. Okay, so it's very important that we come up with a nano medicine or a nano drug. So basically the drugs are being loaded into these nanoparticles, and then they are carrying or they are helping these drugs to travel to the particular uh, cancerous cells only. Okay, uh, then we can also talk about therapeutic antibodies. So over here we have given the example of monoclonal antibody, which are having only the the clonicity or the clonal ability of the antibody is mono, means single only, so they will only divide into a particular type of antibody, or only a specific type of antibody, specific clone of antibody rather. Okay, so again they can be utilized in developing different types of therapeutic antibodies. Okay, so a nice example is given over here. Okay, we are where we are collecting the liver cells, then we are uh, injecting it into the body of the mice or rat. Gradually the rat is starting to develop antibodies against this particular liver cells, cancerous liver cells, and then we are collecting those antibodies and putting it back into the body of the patient who was suffering from liver cancer. Okay, gene therapy, again something very important. Okay, so gene therapy can either be in vivo or ex vivo. In vivo means inside the body, ex vivo means inside the laboratory. Okay, so gene therapy is basically when we are modifying a gene and utilizing that gene to cure any particular disease. Okay, so see, in case when we are talking about ex vivo gene therapy, means which is outside the body in lab conditions. Okay, uh, so in there what we are doing, we are growing the cells in culture. Sorry, we are removing a small portion of the liver. So the, say for example, the patient is suffering from liver cancer, so we are removing some amount of liver cells, then we are growing those cells in culture, and we are inducing the normal genes which are responsible for clotting protein, means the genes which are responsible for coding the clotting factors of the body, and then we are transplanting these liver cells back into the patient. And now the patient, what is the extra thing that the patient is having? The patient is having genetically altered cells which will be starting to produce the clotting proteins, because that was the problem of the patient. Okay, he was having a liver cell genetic defect, so that is the genetic disease, as a result of which he was not being able to, his body was not being able to properly clot blood. And talking about in vivo gene therapy means inside the body. So there we are taking a normal gene which is responsible for normal clotting of blood, and then we are taking some viruses which are acting as vectors for gene delivery, and then we are directly introducing that normal gene into the into the body of the patient. So there can be different targets for gene therapy. So we can target for treating cystic fibrosis. Again, a nice diagram is given over here how we can go with it. Okay, and coming to the challenges of gene therapy, particularly when we are using some viral vectors, it's very important that we make sure that that virus will not lead to any kind of infection, any kind of viral infection inside the body of the patient, which has happened previously. So we have a person called Jesse Gelsinger who actually developed adenoviral infection, and as a result there were various questions that were raised where we should use a vector. Okay, whether it's very much safe to use these viral vectors, or it is very much safe for in vivo gene therapy techniques. But ex vivo is in most of the cases, ex vivo or in vitro in most of the cases is like safe and secure. Okay, so I think that's it. So we will be continuing with the second session of fundamental techniques in medical biotechnology. Please do like, share and subscribe ITLS Academy. Thank you.